Welcome back. In this segment, as I said, I'm going to show you some research that my students and I have done recently, and we just got it published, so I'm kind of excited about it, in a Frontiers, I forget the journal, but I'll fix that maybe. And basically the idea is how we use mycorrhizal fungi, or how we should use mycorrhizal fungi in our thinking as we impose a restoration effort on the landscape. Ignoring it is probably not helping, and I think we showed that in this case. So this is down in Albany, the Albany pine bush. If any of you are from that area, you've probably been there. They're very interested in bringing fire back into the system. Pitch pine forests here on the lower left. Picture I took sort of early spring, and all the pines are down there. As you, there's also oak, a couple of different species of oaks in this area. You can't see any in this site, but there are others. And as spring progresses, you get these beautiful lupin flowerings, as well as a lot of other beautiful flowers. And what they have found is, in the absence of fire, of course they stopped allowing burns, because this is near a mall that's right in the middle of the city, Albany. And so they've been keeping fire out. But turns out that has allowed species that don't like fire to get established. They've been pushing out the pine, and as a result pushing out this lupin. And because of the lupin being less abundant, this beautiful Carnar blue butterfly is far less, or was far less abundant, because it wasn't finding its favorite meal, the lupin. So they want to bring the Carnar blue butterfly. That's the large charismatic megafauna that helped fund my lowly research on mycorrhizal fungi. Little butterfly, okay. So let's look at this work. What they've been doing, I love what they're doing, because it involves fire. I'm not a pyro, but I think fire is important. So from that perspective, I love fire. And so the managers at the Albany Pine Bush are bringing back fire and burning off the vegetation that's gotten in there to compete, I'll use that term, compete with our bishop pine, our lodgepole, not even lodgepole, I'm still on the West Coast, pitch pine. And what they do a couple things. So in the absence of fire, a nitrogen-fixing tree has moved in, locust. It's native, but it doesn't belong there, okay? And so it started to encroach from the edges in the absence of fire over the years. It's been a long time since they've let fire, well, it was a long time since they had let fire burn, because there are houses all over that area. And so the locust moves in and forms this very dense stand that doesn't allow the lupin, doesn't allow a lot of things. There's also a lot of wildlife in that area that doesn't do as well under the encroaching locust stands. So they go in and they cut down the locust. They dig up the roots, because the locust is really smart. It just resprouts. If you cut it down, it has a resprouting root base like your beech does, and it'll just resprout and form an even thicker thicket. So that's not good. But here they're chipping it up, cutting it down, chipping it up, and creating a slash pile, essentially, which they then go back and burn later. And that creates a nice gap. And they were going in and planting, I think there's another fire, yeah, oops, sorry, there it is. So there's a fire in the pine stand. And of course the pine have the serotinous cones, so by and large they're able to handle the fire as long as it's not catastrophic, as long as it doesn't ladder up into the canopy. And so they're very careful when they burn and how they burn, and they're doing it smart. And every year, if you go down there, every year they're burning in small patches. And so those patches are designed in part to run into another patch if you do a burn that allows the fire to go out on its own almost. Now they're being very careful. They're not just like lighting it and walking away. We learned from Yellowstone, we learned from Los Alamos, you've got to manage these things super careful, especially when there's urban environment involved. But they're doing it right. Okay, so after they burn, they go to the site and they plant pine seedlings from New York nurseries, state forest. And by and large, they take. But a bunch of them are failing or were failing and every year. And so there's, gosh, it would be great if we could have more like 100% survival. And they were telling me about this, and I said, well, are you paying attention to the symbionts, to the mycorrhizal fungi? Well, no, we've heard of those. Tell us more. So my students went in there and worked with both the pine and the oak, which are both native, to find out what the mycorrhizal condition is. And Taylor Patterson worked with the pine, so we're going to look mostly at his work. Amy Hewden worked with the oak, and the two of them, that's the paper we just got out with Nahuel Policelli, and I'll talk to you about his stuff in a minute. So here's a site after it's been all chopped up, and it looks pretty horrible. You can see the forest in the background there. The slash pile is right here, so they have used their bulldozer, essentially, and backhoed it, whatever, and cut it all up, got all the roots out, piled them up here, and they're probably going to burn it pretty soon. So there's some heavy equipment over here for chipping, I suspect. There's some others here. It's at a fairly large scale. So, as I said, the pine seedling establishment has been rather spotty. I think actually it's been largely successful, but you always want it to get even better. And that's where we came in. And I saw this, I went hiking out there and I found not this species of mycorrhizal fungus, but a different one called Swillus brevipes. This happens to be Swillus luteus. And I was doing work with Dan Simberloff and his student down in Argentina. We'll run into Dan Simberloff's name in a few lectures, but he came to me, his student, Marta Nunez, came to me and said, hey, we have all these pines that have been planted, and they're supposedly the most invasive species in the world, but not in our area in Argentina, and we'd like to know why. Well, I'm not going to go into that work. We played around with it, came up with some pretty good papers. Then Martin got his PhD, and he went on to a postdoc, and we started looking at other locations, and he got a student named Nahuel Policelli who started looking at some additional questions, and Nahuel is the lead author of this new paper. In this study, my PhD student ran out there, this is now in Chile, and you can see the plantation way back here, okay? And if you dig up the roots of the pine in this plantation, you get Swillus, as that picture showed, but you get some other species, and I'll throw these names out, but don't worry about it, just if you're interested. Hebeloma, probably some Lacarias, some others. And if you cross the road, some of the pine seed is getting out into this plain. And if you dig up the roots here, what Jeremy Hayward showed is you lose most of the species that are back in this plantation. There's about 5 within the first 50 meters away from the plantation are on the seedlings. But there's always Swillus, okay? Always Swillus. And that was 2007. 2015, it's becoming monodominant. It's a type conversion of this high plateau area in the Patagonia region. Chile, I'm not sure if that's Patagonia. The work we were doing in Argentina was near Patagonia. And when Jeremy dug up these roots, he didn't dig up the whole tree and look at every root tip. He dug up a lot of roots from different trees, and every time out here, he got one fungus, Swillus luteus. I saw that, and I saw a lot of other work I've been doing with Swillus from California and Oregon, and I went down to the folks in the Albany pine bush and I said, look, you've got Swillus fruiting naturally. Let's not bring in new inoculum, new species. They may become invasive. I don't like invasions. Let's use the local species. And you've got the right fungus. It will work if we get it onto the seedlings prior to your outplanting. And so they said, okay, let's give it a shot. And we were lucky enough to get some funding. Two different species that we used. Suillus brevipes on the right happens to be from the west coast, but it's out there in the Albany pine bush as well. And then on the left side there is Rhizopogon subroseolus, I believe. We had to identify it by sequence because they're really just little chunks of mushroom stuff. Taylor went out and I collected, he collected, they sent us sporocarps. Taylor then threw the stuff into a blender, ground it up, and just the whole tissue that was producing spores, this surface area here, and the whole thing on these little truffle-like things. And then these are just loaded with spores. And if you put them under a microscope, it's a little hard to see, but we might be able to blow this up. You can just see hundreds and hundreds, literally tens of millions of spores that were in those spore slurries. And then he was able to take those, quantify them, and then pour what was the equivalent of about 10 to the 6 spores per mil over his little germinating seedlings. Okay, so he's just taking the slurry out of here, it's stirring so the spores are in solution, and then he inoculated them. And then used those seedlings to outplant. And here you can see they're growing very happily in the greenhouse. And here are his data. He finished in 2018, just finished. So he's looking at if you sterilize, take that that spore inoculum and autoclave it, and then use that on the seedlings, they get a benefit because it's nutritional, right? It's tissue, there's nitrogen, there's phosphorus. So they do get a benefit. And is that enough to get them to establish? No. They needed the live inoculum, and then you have something like 75% survival versus only something like 30% survival with the sterile controls. So we're convinced that if they manipulate this, and it's not a difficult thing to do, you grow your seedlings like they normally do, but then you pick up the mushrooms, just pick them out, throw them in a blender, make a slurry, pour that water, at some point use that to fertilize, and then use those to outplant, and you'll have better establishment. So we're pretty excited about that. The idea here is restoration, pushing for better survival, better establishment, using the mycorrhizal fungi to help support that. So we're on the way to restoring this pine barrens system. Using fire, we'll get an area that looks like this, patches of pine. There'll be oak in there as well. And they can use the fire to clear out the bottom so that it doesn't get all, doesn't ladder up and cause catastrophic fires. And this open area allows the lupin to get established, which helps the Karner blue butterfly. In contrast to over here on the left side, we have all this locust, very dense, thick vegetation, trees, and not a very healthy forest for what belongs, in some sense, because of the climate and soil types, on this particular habitat or on this particular site. So that brings us to a close for chapter 19. And we're going to move next. Oh, wait, before I go to the next segment, let's bring this to a close with some overview. Remember that succession is a process in which species replace other species over time through a variety of mechanisms, including shading, competition with shading and nutrients, or just tolerance, hanging out long enough to survive and the others die off, etc., right? You know about all of that now. Succession does not always return to the same spot, that is the same plant assemblage or community. You can have multiple types of climax communities. Back to my masters, remember that Doug fir is one community type that is a climax, and that's what we would typically say is the climax. But then a fire comes and knocks it out, and it is not so fire resistant. But there's a soil seed bank in there that after the fire leads to dominance by the chaparral plant called Arctostaphylos, as well as the ceanothus and things like that. And that will be the dominant type for quite a while, chaparral, not trees. And so you have two different stable points alternating. The book talks a little bit about that. And then over time, Doug fir invades back in. The fungi are still there because they share the same fungi as I showed in my master's. And pretty soon you come back to the Douglas fir stand. So multiple climax communities. One is a tree, and that's what we were told was the climax. The other is a chaparral, which I think is equally beautiful and equally stable for a while. Succession occurs through lots of different mechanisms. The Connell slat ear model is really good, but so is the Pickett model. Look at those, and you don't have to memorize all of that stuff in the Pickett model, Pickett Colossa 89, but get a sense. Remember, site availability, species availability, and then it's the interactions between the species that really get interesting. And that I showed you there at the end that you can use all of these ideas to manage a restoration effort more effectively. Rather than just go out to the site, plant trees, and walk away, go out to the site, plant trees that belong there, and walk away. How about plant trees that have the mycorrhizal fungi that they need, and that'll help them get established more rapidly. Okay, so in the next segment, we're going to go into sort of elements and the cycling of elements, decomposition. And I'm excited about this one also because if you think about it, what group of organisms are very important for decomposition? For breaking down wood in particular, it's the fungi. Not mycorrhizal fungi now, back to the ones that you've been learning about in all the other school lessons. It's those guys that are decomposers. That's what fungi do. Well, that's what some fungi do, but we're going to get into those hot and heavy in the next segment. So great, we'll see you next time. Cheers.